Hello everyone. Now I'm starting a new episodes of scientific research by the scientist, and the reference is the newspapers like the Hindu, the Indians, the Times of India, sorry. So as you can clearly see, scientists are trying to interfere in the creation of the God. Are they doing good deed or making the humans future worse? Try to comment and let me tell you the readings of the newspaper. Scientists grow a model of human embryo in the lab, and embryo is an underdevelopment or just developing a human from a cell. So the headlines are as follows that I have already told you. Now let me quickly take you to the readings of the newspaper. Due to ethical reasons and technical challenge, studying human post-implantation development has been limited. As I already told you, it is an ethical challenge for scientists to develop or do a trial on the humans. So the development is limited. The closest that scientists have come to understand intrauterine development after implantation is by using mouse named embryonic stem cells that gave rise to embryonic and extra embryonic stem cells capable of self-assembling into mouse structure stem cell based embryonic models at the post gastrulation stage. That is, humans are not available for their trials because they have to face various human workers who are doing for them. There are various NGOs. If they violate their policies of saving humans, many NGOs working for the humanity, so it is not possible to get trial on the humans and open, so they are doing so unethically. So, but what about the rats or the mouses who are getting victimized by them? Is it good for the development or the future of the humans? They are sacrificing the mouses. So let me another go for the further unmodified nave. It is at this stage that the embryo differentiate into the three primary founding tissue types of the body. But for the first time, researchers were able to extend the findings from mouse to humans by using genetically unmodified human nave embryo stem cells. The results were published recently in Nature, so you can go and further read about it on the Nature. Now, what are the varying methods that scientists have gone through their study? The researchers were able to develop a complete model of human embryo in the lab from implantation into the uterus to 14 days after fertilization. The model mimics the 3D structure and key hallmarks of post implantation human embryos, up with all known features found in normal embryos around the 2 weeks old, known as stem cell based embryonic like structure or SEMS. They were developed without using sperm, eggs, or a worm. These are the key factors that scientists have done so far. That is, without using the eggs in the worm, they have developed a human embryo in the lab with the help of the stem cells and the chemicals. Further, according to researchers at the Weizmann Institute of Science in Israel, the models secreted a hormone that turned a commercial pregnancy test to positive. The authors note that their model recreates the organization of all known lineages and compartments of early post implantation human embryos, including the epiblast, hypoblast, extraembryonic mesoderm, and trophoblast. Unlike previous models, this model shows defining hallmarks of integrated embryo models, including all lineages of post implantation embryo and the structural organization. Spatial allocation. So, these are the main coverings of this news. And further, these are not so important. But the important thing is here is, are we going to accept this that the scientists are doing?